Hi, my name is Maivli Selvan. I am a senior scientist at Gümüş lab at ICA School of Medicine at Mount Sinai. Today I am going to talk about how inherited rare deleterious variant load alters cancer risk, age of onset, and tumor immune microenvironment. Recent studies have shown that inherited genetic variants in certain genes play an important role in cancer susceptibility. These variants are associated with cancer risk in a spectrum from common variants that confer low risk to rare variants that confer high risk. While genome-wide association studies or GWAS have identified multiple common variants associated with cancer, the roles of rare deleterious variants in cancer have not been well studied due to their low population frequency. So our aim is to better characterize the role of RDVs in cancer risk and other cancer-related outcomes, and to understand how accumulation of RDVs in an individual, referred to as RDV load, is associated with cancer risk, age of diagnosis, and tumor immune microenvironment. This study will enable better stratification of high-risk individuals who can then benefit from increased surveillance and early detection programs, thereby improving prognosis. Towards this end, we have performed largest to date jointly processed germline multi-cancer case control association study from existing whole exome sequencing data. This flowchart represents the steps involved in the analysis. We analyzed whole exome sequencing data of around 8,300 cases that spanned 24 different cases from TCGA and around 7,400 controls from multiple dbGaP studies. We first jointly called the genetic variants of all samples together with GATK pipeline. We then performed sample QC and removed related individuals and removed any ancestry-based bias. After variant QC, we focused on rare variants and annotated for pathogenicity using ClinVar database. For increased statistical power, we pursued a collapsing approach and studied the cumulative effects of RDVs at gene level and functionally related gene set level. First, we performed RDV burden analysis, and next, we investigated how the accumulation of RDVs in an individual associated with cancer risk, age of a diagnosis, and tumor microenvironment. We then replicated our findings in an independent clinical case control cohort of 7,700 participants of European ancestry from the ICA School of Medicine BioMe Biobank. To remove any ancestry-based bias, we performed population stratification using principal component analysis and focused on individuals within the largest group, which was those with European ancestry. In our RDV burden analysis at gene level, we confirmed the known associations between cancer risk and germline RDVs in BRCA1 and BRCA2. At gene set level, we studied 4 gene groups related to cancer: cancer predisposition, DNA damage repair, somatic cancer driver, and Fanconi anemia genes. We observed that RDVs in these gene sets increase cancer risk in 2 independent cohorts, both TCGA and BioMe. Next, in our RDV load analysis, we first studied how a personal RDV load in the 4 gene sets affected cancer risk. We found that individuals with higher number of RDVs are associated with increased cancer risk in both discovery and validation cohorts. Next, we studied how the RDV load affects the age of diagnosis in the TCGA cohort. Here we have the age of diagnosis as a function of different groups based on the number of RDVs in an individual in that gene set. We found out that individuals with higher RDV load has significantly high early age of diagnosis. Next, we tested the association of RDV load with the 22 immune cell fractions identified from TCGA solid tumors using CyberSort. The most significant difference between the groups were observed for M1 macrophages. So looking at the M1 macrophages results in detail, we found out that individuals with higher number of RDVs are associated with higher M1 macrophage cell fraction in tumors. Next, we studied how the increased macrophages affected survival. So we performed Cox regression analysis and observed that increased macrophages exhibited significantly worse survival in the highlighted cancer. To give you a summary, we have performed the largest to date case control whole exome sequencing analysis. Our approach has several unique characteristics. We jointly analyze cases together with controls, thus avoided biases associated with using population-level databases as controls. We replicated our findings in an independent validation cohort, and we are the first to highlight the critical importance of germline RDV load in key cancer gene sets. Our results confirmed an extended known association between cancer risk and germline RDVs in genes involved in DDR, CPD, somatic cancer driver, and Fanconi anemia in both discovery and validation cohorts. We showed that increased RDV load in certain gene sets is a potential biomarker for increased cancer risk, younger age of disease onset, and increased M1 macrophages in tumor. Overall, our findings will be used towards identifying high-risk individuals who can then benefit from increased surveillance, earlier screening, and treatment options that exploit their tumor characteristics, thereby improving prognosis. This project was led by Zeynep Gümüş, and other team members are also listed here. This project was funded by grants from Longevity Foundation, Uniting Against Lung Cancer Foundation, and NIH. Thank you all for your attention.